It's been a day. All right, so welcome to day 2 of our Ideas Research Expo. Today we are going to highlight some of our members' research presentations. So we will be having some lightning talks and posters. There's only a few of us presenting today, so we're just kind of going to go back to back to back. Once we are done presenting, you are more than welcome to ask questions, or if you want to email or message the presenter directly, you are more than welcome to do that. So for our schedule today, we are going to start off with Abby, and then we'll go to Brian, and then we'll go to myself, and then we'll go to Matthew. And then here is the names of the research that they will be presenting today. So I'm going to go ahead and stop sharing my screen. And then I'm going to go ahead and give the power to, let me make sure everybody can share their screen real quick. Okay, cool. We're going to go ahead and let Abby start.
Okay. So my research currently focuses on studying high uric acid readings amongst patients with systematic lupus, erythematosus, and kidney failure. What is uric acid and how is it related to the kidneys? Uric acid is an end product of purine metabolism in humans. It's largely excreted by the kidneys. Uric acid levels rise due to the reduction in the glomerular filtration rate, which is also known as GFR, and it causes kidney injury via endothelial dysfunction, inflammation, and oxidative stress. I pulled this image that I created using BioRender from my poster presentation that I presented at Biosci last semester, and it shows the blockage that can occur due to the buildup of uric acid and the inflammation that it can that it can create in the glomerulus capsules. So I know this is very wordy. The methods that I used were primarily from the All of Us research database. We created a cohort of SLE patients with kidney failure and high uric acid readings. I use the parameters for determining a high uric acid reading as uric acid serum readings to be greater than 6 milligrams per deciliter, and for urine to be less than 250 milligrams. Of the patients with SLE, 632 of them had received at least one high uric acid reading, and 328 had received at least one kidney failure diagnosis. I also ran a comparative cohort, which was created just on patients who had received high uric acid readings. The total count was 20,012 patients. And of those patients, only 6,836 of these participants had at least one diagnosis of kidney failure. So here is my pie chart demonstrating what I found in these two cohorts. As we can see, patients, when you add SLE in as a comorbidity, it actually increases the number of patients that have kidney failure versus no kidney failure. And if you look at patients with simply kidney failure alone, they are significantly lower in the spectrum of having at least one kidney failure diagnosis. We also did a chronological graph to look at the diagnosis time periods for all three comorbidities. And as we can see, high uric acid readings and systematic lupus diagnoses start around the same time frame. And the high uric acid readings always maintain above the number of patients that are diagnosed with kidney failure. It's also important to note that it starts before the diagnoses of kidney failure. So this helps strengthen the idea that high uric acid readings would be a great early marker for kidney failure diagnoses and could potentially serve well on the panel screening that systematic lupus patients go through pretty much monthly to yearly. And the conclusions we came to, Patients who suffer from SLE and kidney failure are more prone to have the high uric acid readings at 52% of them when compared to patients that suffer from kidney failure alone, which was at 34%. High uric acid readings and SLE diagnoses occurred around the same time, but kidney failure diagnosis started a later period. As I mentioned, this helps strengthen the theory that uric acid would be a potential early marker. Current lack of research in how high uric acid levels affect the function of the glomerulus and the functionality of cells responsible for filtration. I tried to find research on strictly glomerular endothelial cells, and I have been unable to find any. So we're unsure how uric acid in different concentration is actually damaging or not damaging these cells. And I plan on doing a future project for my Barrett thesis studying this factor. Current uric acid lowering treatments also pose a risk to those with autoimmune disease due to their reactivity. We found a lot of patients that were taking the uric acid lowering medications that it exacerbated their condition for lupus. So we should look more into feasible treatments for these folks that are autoimmune disease diagnosed. Future directions. Future studies should be conducted on how high uric acid levels affect the glomerulus and the cells responsible for blood filtration, as I mentioned prior. Research should be done on the effectiveness and reliability of current treatments used for high uric acid levels in patients. As I mentioned, they exacerbate the symptoms that SLE patients go through, and it's already very, I don't want to say, how do I want to say that? It's very weight bearing to have SLE. It causes a lot of reactions that are inflammatory with the skin, other organs. It's already a difficult disease, and we should be looking at treatments that don't make this disease worse for them. And that's basically what that says for right there for this tidbit. So yeah, looking at the medications to lower uric acid for these patients would be very feasible line of research in the future. And finally, we would like to acknowledge Solar and the School of Life Sciences for providing this opportunity to do this research, as well as the All of Us research program. All the data that was presented in this presentation today was compiled from the All of Us research program. And if you'd like to view my references, you can scan this QR code. This QR code will take you directly to my sheet of references for any papers that were used to compile this research.
Yay. And then Abby this morning in our lab meeting presented another finding that she found about uric acid release, which was really cool. So if you would like to see her presentation on that or have her show her slides, you are more than welcome to message her about it. Any questions on her research? Comments, concerns? All right. We'll go ahead and move on to Brian.
Awesome. Let me just here. What? There we go. Is it showing the full screen or there we go. It
Yeah,
changed.
perfect.
Yeah, it was going a little slow earlier while I was testing it out, so. All right. So my name's Brian, and I'm currently working on a study, or I'm in preparation for a study called Beyond the Battlefield, Examining Stress Progression and Resilience in Combat Instruction. I'm focusing on a community within the Marine Corps, combat instructors. It is the second step of your initial training after you join the Marine Corps. So you go to boot camp. After your 13 weeks in boot camp, you'll come to Marine combat training, or you'll go to infantry training if you're an infantryman. And then you'll spend about 29 days there, depending on which battalion you go to. If you're Marine combat training, you'll be there for about 29 days. And then you'll go from infantry training to your MOS training. So if you're a mortarman or a machine gunner, you'll spend about 3 weeks in the initial training, and then you'll go on to however many months of your follow-on training. A little bit about myself. I was in the Marine Corps for about 8.5 years, and I was a combat instructor, so that's why this is very important to me. Right now, it's just myself working on this, and a friend of mine who is also a Marine. He's over in Oregon right now, and I've been working with Dr. Ruth as my PI. Reached out to him one day, and I was like, "Hey, as I'll talk later in this presentation, I'm going to be looking at like 100 participants. That's a lot to be interviewing by myself. And he's getting a minor in anthropology. So he was fully on board. He's ready for this. So it's just the two of us. And for myself, I'm only doing this as an undergraduate, but this study will be carried out in multiple phases. This is just the initial phase to gather some intel. And then when I move on to my master's, probably next fall, I'll carry it on into the second phase and hopefully lengthen it, because right now we're just looking at 3 months. And then in the second phase, I'm probably looking around 1 year to about 3 years. And for the background, let me just move this out of my screen. There's about 300 combat instructors at any given time. There's one on the West Coast at San Diego and one on the East Coast. I'll be focusing on the East Coast where I was. And instructors can vary. Like I said, there's the infantry battalion and there's the Marine combat battalion. If you're infantry, you're sticking with infantry. They want their instructors to be infantry guys. But if you're going to Marine Combat Training Battalion, then you've got admin guys, you've got mechanics like myself, drivers. Anything that isn't infantry is going to go be an instructor at MCT. So you have a lot of diverse background. And a normal tour is about 3 years long. So you're leaving your main job for about 3 years. The lowest rank that you can be is a corporal. Normally people get promoted to corporal around 2 years in, unless they're meritoriously promoted. So you're looking at a 2 year seasoned Marine up until you've been in for about 10 years. So anywhere in between 2 and 10 years, you could go be an instructor and leave that for about 3 years. So you have all this background from your job, from your home life, if you're married, if you're single, living in the barracks. And I wanted to look at all these things and see what they contribute to stress as the job progresses as a combat instructor. Like I said, we're looking at 100 instructors from across the School of Infantry East. This includes the advanced schools. So you've got infantry training battalion or advanced infantry training battalion, infantry training battalion, and Marine combat training. But you also have instructor school. So before you become an instructor, you have to go to a 9-week course. I think they might have lengthened it to about 12 weeks now. So it's about 9 to 12. I know when I was leaving 2 years ago, they were introducing a new shooting package to train the instructors how to shoot their rifles in a, say, like a different way. And that takes up to about 2.5 weeks. So I'm looking at the stress you get when you come into the school, when you leave the school and go become an instructor. And then I'm also looking at prior combat instructors, because this is going to focus on what your stress looks like as you're coming into combat instructing. And then phase 3, phase 2, we haven't really decided yet. It's going to look at 6 months after or any period after, right? For phase 1 right now, I'm going to look at anyone I've known. So the people I know, I'm going to ask if they want to participate. Myself, I might look at myself, because there's a lot of different experiences as you get out, because the way your brain is wired changes. Everyone thinks of PTSD as combat related when it comes to military, but just doing the job every day, day to day, it's still a 24-hour position. Some days throughout the training cycle, you're going to be in the field, in the woods for 2 days at a time straight, and you get 4 days off in between a cycle. So you're going 29 days, 4 days off, 29 more days. So all of that's going to take a toll on your stress, on the resiliency. It might give you some PTSD from it. So I'm looking at everything that's going on and then how it changes when you get out. And leading into that, as you get out, a lot of people have insomnia, they have depression, they have anxiety, alcoholism, anger issues. Um, Myself, I suffer with insomnia because of this. Um, I know people who have anger issues well past, like we're talking like a year after after they left, and they're still acting like they're working with students, like they're brand new privates, and they're talking to seasoned lance corporals, seasoned PFCs, like they've been in the Marine Corps for 3 years. They're not a student anymore, and they just revert back to something happens, they snap, and they revert back to being an instructor. So that's what I'm looking at. Where the change is it in the beginning? Is it in the middle? Is it when you're 3 years in and that's all you're used to? Where the change exactly is while you're an instructor for that stress. Like I said, I have friends and family that have faced the same struggles, sorry, as myself. And we aim to use this data to create better stress management programs for not only just combat instructors, but as I'll talk later, I have a couple other studies that I'll show with drill instructors, recruiters. This can also lead into Marines within the fleet or within the Army or the Air Force. Just better stress management than what's already been provided, because as it stands, you can objectively say that it's not working. Everyone's still stressed. Everyone's getting PTSD. We haven't actually been in a war since the early 2000s. Sure, you could say the war on terrorism has gone on and it just ended a couple of years ago, but we haven't actively been in combat for years. But there's still high rates of PTSD, high rates of suicide. And what we want to do with this study is get to the bottom of it right from a small community here in the States. Like I said, that relevant data. There was a study with Army drill sergeants, and then there was another one that kind of stemmed where I stemmed from on Marine drill instructors. Both of them looking at the same thing of your initial boot camp instructors. What are the effects of being an instructor? Who's walking away with anxiety? Who's got burnout, alcohol abuse, off-duty aggression? And both of these studies are really good. I know they're linked in this PowerPoint, but you can easily look it up online. And they just nitty gritty in depth with where it started to stem from and what became of it. The one for the Marine drill instructors, um, Carrie Fosher, she's now the director for the Marine Corps Human Research Prevention Program, and she's the one that actually pointed me to both of these studies. And she's a big advocate for my own study because she focused on the drill instructors when she did her study, and she didn't think about the recruiters or the combat instructors. And so it would be a big, kind of like a big deal to get this study done, and that's what I'm aiming for. Unfortunately, right now I'm just in the IRB process, but this is where I hope to be. Somewhere down the line, hopefully someone will do a presentation with my own data. And then this is the bibliography for both of those studies. But with that, I believe I'm done. If anyone has any questions or anything.
Comments, concerns. That's really cool. That's a very, Jeanette raised your hand. Go ahead, Jeanette.
Sorry. I'm wondering if you're only doing interviews with the Marines themselves or also with like their families.
So right now I'm just looking at the Marines themselves. That's you bring up a good point, and I could add that into it. We're still trying to figure out how we want to do the interviews. Um, right now, the way it looks is that we're going to do group interviews for the first phase. I keep saying phases, but what we plan to do, we plan to start in September, and then we're going to do group interviews for the first week, and we'll come back in a month and do individual interviews. But
I mean, 100 sounds like a whole lot, but at the same time, like I'm not sure how open Marines are really known to being, if that makes sense.
you're right, we're not. We really aren't. But I know the group that I'm looking at, and that's kind of why I wanted to lean into this group because I've been there. I not just as a Marine, but like I've been in this community and I know some of the people that are still there that might wind up being part of this. And
That'll
anything
help.
to get a better voice. Because we're more looking for the work related than home. Like home plays a part in it, but we're looking more towards like the work stress and what comes out of
Mhm.
that. And it is very um self-advocating, I guess. Because the way it looks now is that the command focuses on the students. No one really takes a look at the instructors, and to have someone come back and pay attention to them, I think they would be very open to at least trying to see what could come of it.
That's great. Thank you.
You're welcome.
Good job, Brian. That was fun. That was a good one I'm excited to see where this goes and see if people continue after you guys finish you finish your stuff and then someone continues with it. Cause I know I've seen you talk about it in the Discord. So I'm
I
excited.
hope so. With everything together, like the way I'm looking at it, with three phases alone, it could take the next like 6 years.
Oh, wow.
So hopefully someone takes it and runs with it with another branch or something.
I think they will. I think it's interesting enough and it's something that I think needs to be studied as well, especially because PTSD can happen in multiple ways, not just how a lot of people think that it can happen.
Yeah, and I meant to say it during the presentation, but I spoke with Dr. Trumbull about it, and you could take it. I'm more focused on the behavioral aspect, like does your job pay anything into your stress? Does your home life pay into your stress? Because obviously, if you're an administrator and you're working in an office day to day and you're just pushing paperwork, you're not going to have the same stress levels as a mortarman who's going to field exercises, launching mortars into training, or going into combat launching mortars. So just that alone and bringing it to being a combat instructor is going to be immensely different. Um, the environment that you're in before you come compared to what Dr. Trumbull does. He focuses on cortisol levels and all that, and he thinks that the behavioral aspect would be really good. And if either I start a different study or if someone else came behind me, like you said, they could use cortisol levels to get an in-depth look at their stress and the work environment and everything like that.
Nice. Ah, perfect. Thank you for sharing your research proposal and your direction that you want to go. It's very interesting. So, all right, now it's my turn. So let me go ahead and share my screen. So I'm going to be sharing my poster that I presented at Biosci this past fall, and then kind of give how this research project kind of is leading towards where I'm going with my capstone project, which is a literature review right now. So for those of you that don't know, I am currently studying how processed sugars in US infant formula are affecting infant and toddler development to where they are developing diseases early on in age that will eventually also influence other diseases later in life as an adult, such as colon cancer, ovarian cancer, type 2 diabetes, Alzheimer's, ADHD, autism, anything that's related to the gut, and how the processed sugars either speed up or slow down those processes. So for the past roughly 2 years, and this is kind of the brainchild of Dr. Holchek and I, we have currently been looking at previous research and previous literature. Over the summer, she did have me write a literature review on specific bacterial, animal, and human cell lines that would be used to help create an experiment that I basically created for BioSci in this poster over the fall, thanks to the Sol +1 and solar. Basically what we did is I decided to use a bacteria called E. coli Nissle, which is actually in our gut, and it actually regulates glucose through the liver and the insulin receptors in our bodies. So basically what we wanted to see was how the different processed sugars in infant formula, such as Similac and Enfamil, affected their growth and proliferation based on a certain environment that we created. So during immersion last year, we basically set up 2 different types of experiments. One was to test how E. coli Nissle functioned in the process or in the presence of processed sugars in a salt environment that mimicked our intestinal environment. So we basically created 4 different agar plates. We put the E. coli on there with the processed sugars in the agar. And we tested Enfamil, Gentle Ease, Similac Advance, and regular Enfamil, and then Similac Alimentium, because those are the more common baby formulas that you'll find in the US. And we found that even though it's a salty environment and E. coli is known to not thrive in salty environments, we found that if you can see up here in the, whoa, that's not what I wanted. Hold on, we're gonna actually go into present mode. Let me go into present real quick. There we go. Play from start. There we go. Okay. So you see in the upper right hand corner, you can see that with the Enfamil Gentle Ease, the colonies actually proliferated a lot more than the Similac Alimentium. And we are thinking it's because it's due to the amount of processed sugars that is in the Gentle Ease. We did find that the more sensitive formulas have more corn syrup solids, more added glucose, and more just added processed sugars in general. So we're thinking that the processed sugars kind of helped E. coli Nissle grow over time, and that the amount of processed sugar also played a factor. Additionally, we also did a growth rate. It was a 9-hour experiment where every hour we basically created a minimal medium that was with our baby formula, our M63 and 0.2 NaCl solution. We let those sit in the incubator for overnight so that you can get the colonies of the E. coli to grow. And then every hour we would let them ferment. We would take samples from each one. We put it on the plate. We'd let it sit. And then we sit as soon as we were done after 9 hours of letting our solution just kind of ferment and incubate over that time. We let them sit for a total of 24 to 48 hours. And we came back and we counted the amount of cell growth when they are exposed to the processed sugars over a specific amount of time. So as you can see, they all kind of started very, very similar. And then they kind of, as you can see, over the 8-hour, 9-hour time period, it increased very, very rapidly. So it means that what we think is that the processed sugars are aiding in the overproliferation of these E. coli cells, which technically they're beneficial bacteria because they help with glucose regulation. But we also know that overproliferation can be bad. So our next goal is to see how eventually this would affect in animal cell lines and human cell lines. So now that takes me to my current project that I'm working on, which is a literature review on how the processed sugars in infant formulas affect cellular, metabolic, and immune functions within infants 0 to 12 months. And currently we have found, or I have found within the literature review, is that infants who are exposed to processed sugars from the time they're born up until 12 months are experiencing higher rates of cellular integrity dysfunction and allowing more flow of pathogenic bacteria into the bloodstream, which is then causing an activation of pro-inflammatory cytokines, leading to the development of more autoimmune diseases and even metabolic, I can't talk today, metabolic diseases such as IBS and type 1 diabetes. Additionally, it was shown that a lot of formulas don't actually include the human milk oligosaccharides, which is found in breast milk. It's the typical natural sugar that mothers produce in their breast milk. It also varies based on diet. So a mother can have more HMOs comparative to another one based on their current diet, how well they're hydrated, and then just culture also plays a factor into that, because a lot of women in the US and Europe formula feed now due to the culture, whereas you have other cultures such as they're Africans or other like rural communities where they more focus on breastfeeding. So we found that not all formulas have that, but if the formula did have HMOs and processed sugars in it, it was shown that the HMOs can potentially negate the disruptions that the processed sugars are creating within our gut, leading to less disease development. And also, too, because of this, it is affecting the gut microbiome, because these processed sugars are causing an overproliferation of sugar-mediated or pathogenic bacteria instead of the beneficial bacteria that will help keep a balanced environment. So it's really interesting to see how the different cultures and everything also plays a role into this, because there are 4 specific single nucleotide polymorphisms that play into how different ethnicities and cultures process sugars. With one SMP, there is a higher risk of hypertension and cardiovascular disease development in Americans and Europeans comparative to Asians and Africans, whereas it was also shown that Hispanic infants who consume lactose reduced or lactose free, so soy milk or goat milk formulas, actually had a higher increase of weight gain and obesity, while also experiencing more insulin and glucose resistance based on the variation of the SNP that was in their genetic DNA. So with this literature review that I'm working on for my capstone, my future directions is to come up with a experiment or a research project that builds off the one that I did for biosci, but also including the ethnic factor and the different polymorphisms that can affect sugar digestion within our bodies, especially because a lot of the research that I'm seeing is on rodent models or human adult cell lines, and you're not getting the same results as if you would test this on infant cell lines, because human cell lines have had that time to mature and develop and gain the resistance that they need, whereas we really want to see where these processed sugars really affect infant cell lines and how we can hopefully change the composition of American infant formula to where we decrease the processed sugars, maybe add more HMOs, but still make it to where they are still able to benefit from formula, because as always, fed is best, but we're seeing so much disease development and diagnosis earlier on in life based on the food and the cultural and societal norms that we see here in America and Europe. So that's my research. Any questions, comments, concerns? That's been a lot. Currently. Yes, Jeanette.
Sorry.
No, you're good.
You said that those are the formulas that are most common in America.
Mm-hmm.
What formulas, like would formulas that are more common in other countries, would they have different levels um like
The processed sugars.
yes, I'm like
Yes.
my brain is
I
breaking.
did find an article that talked about European processed sugars. And while they do have less than what the Americans have, there's still a good amount of processed sugars. And we also are finding in 2 of the primary studies that I am using that when you look at the nutritional labels, the amount of sugar that's actually listed on the nutritional label is not the total amount of sugar that's in the infant formula. And they're finding that in American infant formula, it's an 82% difference from what's on the label to what they've found in a lab. And in the European formula, it's about a 65% difference.
Interesting.
So it's very misleading to a lot of parents, and because some women can't breastfeed, some infants are allergic to cow's milk, they're allergic to the lactose, so they have to go on soy or goat, and typically soy and goat have a lot more processed sugars in them. So not being able to know exactly how much sugar is in the formula is very misleading, leading to overconsumption, which is then in turn influencing how the gut microbiome develop and causing the increase of disease development, neurological development, immune function. It actually affects both the adaptive and the innate immune system in different ways. And affecting the innate immune system is not really good, especially when it activates a lot of pro-inflammatory cytokines and toll-like receptors. So it's more of seeing how much sugar and how these processed sugars affect us, so we can reformulate our formula to make it more beneficial for infants than it already is.
Thank you.
You're welcome. All right. So we're going to go ahead and go with Matthew.
All righty. Let me see if I do this correctly here. All right. Is everyone able to see my screen there? Well,
Still loading, but let's see. Yay. There we go.
perfect. Okay. So the research that I have been working on has been mutagens pertaining to bioengineered food and then the related pesticides that are used with those products and crops and their links to disease. And so this actually, this research has been performed with solar. I'm actually a 4+1 student in the School of Life Sciences, and I have completed my undergraduate degree in biomedical sciences, and so my master's is in biology. And so this is what I completed last semester and is going to lead into this next semester, which is going to be here in a couple of weeks, or I guess next week, where I'll be coming down and working with human lung cells and some of the pesticides that are actively sprayed. And then if anybody wants to contact me after this, there's my email there at the bottom. And so, why study bioengineering, pesticides, and mutagens? Well, I'm from Washington State, and so I'm from Whitman County, and so we produce more wheat than any other county in the United States. And so, consequently, because of our environment, we live in a desert, a semi-arid desert, and so we don't really get a lot of rain. And to combat that, we use a lot of genetically engineered wheat. And so over the past couple of years, working with the United States Department of Agriculture and Washington State University, I was able to directly work with some of these farmers and get some hands-on experience with what they're actually doing. And then that translated to my lab experience here at Arizona State University with Dr. Holacek's lab and solar. So that then begs the question, what is a mutagen? Well, mutagens are chemical or physical agents which induce changes in our DNA. And so these mutations contribute to certain diseases, like cancer. They can also ultimately lead to mental decline and increased rates of aging. And so in order to really understand genetic engineering and what processes are being done to crops, the first semester that I came down to Arizona State University, we actually induced these genetic changes in plants with agrobacterium infiltration. And so we artificially induced these changes, and then we used different organism DNA and we combined that into the plant genome. And then once that bacteria was able to really insert itself into the host genome, it's able to replicate itself, and you now have a transgenic plant. And so you can actually see this picture here is the process that me and my team did for that agro infiltration process. And so that then led to the next semester, and we were testing products to test for the presence of genetically engineered DNA. So we were looking for different proteins that would basically be expressed. And you can actually see here on the gel electrophoresis results that we actually have some DNA where there isn't supposed to be genetically engineered DNA. And so we did a little more research, and it actually turns out that there are viruses like the cauliflower mosaic virus, which are able to naturally insert themselves into plant genomes in the wild, and they infect a wide variety of species. And so what that ultimately means is it's much more difficult to be able to detect the presence of genetically engineered products versus populations of plants that have been naturally infected by this virus. And so that led me to look into a lot of other aspects of genetic engineering. And one area that I was particularly concerned about is as we eat these genetically engineered crops, what is actually happening? And there's a really good case study that happened in 1999 by Arpad Pusztai. And he actually fed rats different diets in order to determine whether or not genetically engineered crops are actually producing any adverse effects in animals. And so he used rats, and so he compared normal potatoes to potatoes with added lectin, because at the time in the 1990s, it was argued from manufacturers that lectin was actually causing disease in animals, not the genetically engineered product itself. And then they also fed a different population of rats only genetically engineered foods. And so the mice that were only adversely affected were the rats that were fed the genetically engineered crops. And so at day 10, the rats were harvested and analyzed, and then they also had rats that were harvested at day 110. By day 10, for the genetically engineered crops, there were noticeable differences in virtually every single organ system after that initial 10-day period. And you can see enlarged pancreas, partial atrophy of the liver, and you can actually see here on the left-hand side, especially in the top, we actually have the stomach lining, where you can see in the first image, there's very stable cell growth. All of the cells look very uniform, whereas if you look at image B on the right-hand side, you can see prolific cell growth. You can see that the cells are swollen. They don't look very happy, and they look stressed. And it's also the same case if you look at the bottom two pictures as well. If you're looking at the normal non-genetically engineered liver, and you're looking at the genetically engineered, the rat that ate the genetically engineered crops. And so that also led me to what I want to do this semester, which is looking at genetically engineered foods and also looking at glyphosate. I've looked a little bit at the actual genetically engineered portion of things, but I wanted to start looking at, for example, Roundup. It's one of the most prolifically sprayed products, at least where I live, and you can actually see here on the map up over in the left-hand corner. Washington State, you can see Whitman County, and there's one other county that are the darkest blue counties in Washington State. And that does play a big role. We also know that this is a likely carcinogenic chemical. There are a lot of legal cases against this particular chemical, and it's really controversial. GMO crops also account for 56% total use of the chemical, and it's a $9.14 billion industry. So there's a very, very strong business incentive to use this product. We know that there are ecological impacts on plants and animals, that there are microbe and plant relationship disruptions, plant-predator relationship disruptions, and aquatic environment disruptions as well. And so, culminating this all together, there are a lot of different issues with reporting with genetically engineered products and the chemicals that we're using. Industry and government currently claim that there are no adverse effects, and GMOs and glyphosate are safe for regulated use. Independent research, on the other hand, is showing that GMO and glyphosate is actually not potentially safe. And the consensus really isn't clear. And so we need more research for acute and chronic implications of genetically engineered crops and those chemicals. And so what I aim to actually do this next, in the next couple of weeks, when I come down to campus, is to actually perform a cytotoxic assay and determine what the acute level of toxicity is for Roundup and glyphosate. And then I actually want to introduce that into a cell system with MRC5 cells, which are human lung cells. So that way I can actually see what is happening as this chemical is being introduced at a lower level when farmers would typically be spraying it, and over the course of a week, what is going to happen to those cells. We know that glyphosate typically has a 2 to 3 day lag time in terms of exposure and effects in the human body and in mammalian systems. And so being able to mimic that in the lab is going to be a good next step in order to actually determine whether or not glyphosate is actually a problem, or if it's more related to the adjuvants and the things that they are combining with glyphosate for those products. And so there's a QR code if anybody would like to scan for my references. And then here are my acknowledgements. Thanks, everyone.
Yay. I'm so excited to see what you get next week when you present during the solar seminar and see the results. Like if you get results.
I'm very excited. It could be one of those things. I mean, it is, you know, it's a cell culture system. And it could be that nothing will happen. We just don't really know. And that's one of the nice things about performing cell culture experiments is you get a good snapshot as to whether or not if it's something to do with that particular cell system, or if there's something overall physiologically that's going on. And so this is going to be a really good, I think, next experiment to be able to see whether or not it actually is related to either the physiological human system, or if it's related to the adjuvants, or if glyphosate itself is cytotoxic. So there's going to be a lot of, I think, good, insightful results no matter what happens next week.
Yeah, I'm looking forward to seeing that presentation. All right. So next up, we have a last minute presentation by Susan.
Hello, I'm going to talk about molecular mimicry. So molecular mimicry is when there's a structural or sequence that is similar from an exogenous peptide and a self-peptide, and that is called a mimotope. These mimotopes cause a cross-reaction with T or B cells and leads to a breakdown in self-tolerance, triggering an immune response against self-antigens. This has become a prominent theory for why those genetically exposed to autoimmune diseases this is how the pathogenesis occurs. So molecular mimicry these mimotopes occur from symbiotic microbiota, and pathogens. So the ones in our gut and other areas, like our skin, respiratory tract, and then invading pathogens like Epstein-Barr virus and SARS-CoV-2. This has been hypothesized to occur that microbes use molecular mimicry to disguise themselves from being caught by a host's immune system. And there is a delicate balance, as this can lead to an excessive autoimmune reaction, which can cause a pro-inflammatory cascade and potentially causing the host's death, which would not be good for the microbe or pathogen. So cross-reactivity. Cross-reactivity is one receptor can see multiple different antigens. And this is on T cells from our immune system, and they are able to see so many different foreign peptides to protect us from infection. However, this is what is to, and then this initiates an immune response against those foreign peptides. However, due to molecular mimicry, it can also trigger an immune response against our own cell. So we know that RA is a combination of both genetic and environmental factors, specifically with the HLA-DRB1 allele, which is shown to be a high-risk allele for those later developing rheumatoid arthritis. It is believed that this allele type determines what peptides are presented on CD4 T cells. All right, these are from bioinformatics, multiple viruses, bacteria, as well as microbiota that lives within us that can become pathogenic in certain cases, that has been found to be related to molecular mimicry and thus causing an autoimmune response and an autoimmune disease within individuals. This is for RA, and these are for lupus. Most of them tend to be different pathogens, though there are a couple that are similar, like Epstein-Barr virus. The gut microbiome has been extensively researched, involving autoimmunity and the pathogenesis of rheumatoid arthritis, specifically Prevotella copri, which has been shown to appear in new onset RA patients, as well as in experiments. It has been shown to induce arthritis in mice. However, there are still a number of areas that still need to be researched. Specifically, Mycoplasma fermentans has been shown to be a bacteria that's from the urogenital and respiratory tracts that has been shown to become pathogenic and due to molecular mimicry and trigger an immune response. So some further research in RA and lupus, there tends to be a greater ratio of women versus men. And it's interesting that there's been little research done in urogenital or vaginal microbiota. The end.
Yay. So are you hoping to, with your research, use this to hopefully work on a future project?
Yeah, yes, I would like to find a cell line to see if I can stimulate an immune response via molecular mimicry from either a bacteria or a virus that commonly dwells in that area or causes infection in that specific area.
Ooh, I'm loving the fact that everything's kind of coming back to gut microbiome. Like, we're seeing a lot of research coming and trying to understand our gut microbiome because so much is now linked to its development and how it affects so many different types of functions within our bodies. Yay. All right. So that concludes our research presentation for our Ideas Research Expo weekend. I really hope that you guys enjoyed both the presentations from yesterday and today. Both recordings will be up on our YouTube by tomorrow, Tuesday, the latest. And then I want to say thank you to those of you that presented. We really, really appreciate it. We love to see when our members take the initiative, get really involved in a concept or research that they're really passionate about and love sharing it with our members in hopes to inspire them to continue and follow in some of the footsteps of our members. But with that, I'd like to thank you for coming and y'all have a good day.